Hello, everyone. Thank you very much for coming today. My name is Charlie. I'm the chair of the Imperial College Computational Biology Society. It is my pleasure to welcome you all to this joint seminar between SynBIC and IC-CompBioSoc. This is our first joint seminar together, and I would like to thank all of those at SynBIC for collaborating with us to make this happen. Today's lecture will be with LabGenius, an innovative startup based here in London that works at the intersection of synthetic biology, computational biology, and AI to develop new protein therapeutics. LabGenius was founded in 2002 by a team of Imperial students and has since then grown considerably in size and gone on to raise tens of millions of pounds in funding. I would like to hand you over to Brandon, who is the president of SynBIC, and will be introducing our guest today. Thank you.
Thank you, Charlie. Before I start introducing our speaker, I would just like all of you to know that if you guys have any questions at all during the talk, just write them down in the chat and we'll go through them at the very end. So our speaker today will be Harry Rickerby. Harry completed his MRes in Systems and Synthetic Biology right here in Imperial, where he served as a founding member of LabGenius. He currently serves as platform architect and board director for the company, and in this role, he is focused on improving the scalability, traceability, and reproducibility of LabGenius's protein engineering platform. We are delighted to be able to hear from Harry regarding the work currently being done at LabGenius at the interface of synthetic and computational biology. I will now hand over to Harry.
Cool, thanks very much. It's really, really nice to be here and amazing to see so many people on this Zoom call. I'm really excited to tell you a bit about some of the work we've been doing at LabGenius today. And yeah, we'll get into it. I'll share my screen now. Just do this. Cool. Now go away, present. All right, can you see my screen? Yeah, awesome. Okay, cool. So hello, everyone. Yeah, so this talk, really happy to be giving it today. I was scheduled to give it quite a long time ago. I think it was like sometime late last year, and something came up. Unfortunately, I was not very well, and so had to miss it. But I'm really happy to be able to give it today. I'm so happy that there's so many people here to listen. So yeah, just basically a quick intro to us as a company, first of all. Really what we're trying to do is to work, we work in the area of protein therapeutics, and we're trying to combine a bunch of different technologies together to really sort of fundamentally change the way that protein therapeutics are discovered. And that means combining synthetic biology with machine learning, computational biology, robotic automation, and really trying to systemize and really kind of streamline the process of discovering new therapeutics. Just a quick intro on the company, where we are. So we, as Brandon mentioned, we came out of Imperial College. So I did my master's at Imperial about 6.5 years ago now, and we worked in the basement of the Bessemer building for a little while before we moved out. We've also been based in the in the basement of Somerset House, somewhere further up the river in this photograph, and now we're based on the site of an old biscuit factory in Bermondsey, where we have our lab and our offices. Obviously, with everything that's been going on last year and this year, we've had to cut back on some of the number of people who go in, but we've still managed to keep things ticking over during COVID, getting all of our key sort of experiments done and developing our platform. So yeah, as I mentioned, what we're really trying to do here is to build a protein engineering platform. So we're trying to make really systemize the process of discovering new proteins, and particularly new protein therapeutics, by combining machine learning. So obviously a very exciting area of research in the moment with automation and synthetic biology. And while machine learning is kind of the most sort of sexy new technology that that we play around with, it really couldn't. It's kind of irrelevant in without those other aspects, without synthetic biology, without systemization, without automation, because fundamentally machine learning relies on high quality data at the end of the day. And before you can do any of that stuff, you need to generate good quality data. And that's where all of those other things come in. We are staffed by a team of about 45 people, and we're growing at the moment. And we combine, we have lots and lots of different backgrounds, people from, we sort of started out with a lot of academics, biologists, predominantly, and then computational biologists. And since we've been growing, we've been adding, we have a software team, we have data scientists, we have automation engineers, people from tech, people from we have a guy who worked at Volvo, loads and loads of different disciplines in different areas. And that's kind of key to who we are as a company, is combining all of those different fields together. Our focus, as I mentioned earlier, is really on the discovery of new protein therapeutics. And so just for those of you who aren't aware of the process of drug discovery, I'm sure that a lot of you have probably learned more about it this year and last year, but there is it's a very, very long and drawn out process. It starts with target ID and target validation, i.e. What is the specific molecule that you're looking to develop a drug against? And that relates to the disease biology. Then there's a whole bunch of steps relating to drug discovery. So once you've selected your target, you then need to develop assays in order to figure out whether your drugs are going to work or not. You need to generate hits. You need to generate some new molecules that we think are promising against the target that you selected. You then sort of start to optimize those hits into leads, checking how well they perform against not just do they hit the target, but also do they look like they're going to be safe? Can we produce them at high enough yields? All of those kinds of questions. And then there's typically some kind of optimization process after that. And then that's kind of where currently our focus ends. So after you have your lead molecules that you've discovered, you start to put them into animal models, seeing how they do in vivo, and then you start to go into more of the clinical development work. So starting to put them into humans and seeing how they do in that context. Our ambition as a company is to not just live in this space that we live in today, but also to start to expand both upstream and downstream of this. So we're actually starting to think about doing our own animal model validation on some of our molecules this year. And then as we grow and as we scale, we'll want to take our own molecules further and further towards the clinic. But right now, really, we're sort of focused on the discovery stage. So basically going to give you an overview of what this means. So when we talk about machine learning driven protein engineering, what does that actually mean? What does the process look like? What are the benefits? What are the drawbacks of taking this approach to discovering new proteins? Proteins as a class of molecule are incredibly powerful, and there are a ton of different molecules that exist already that we've discovered that have been used to treat a whole bunch of different diseases. And while I say that there are a lot of molecules out there, there are an almost infinite number of molecules that could possibly exist for treating diseases. And actually the number that have been approved and taken to market is relatively small in comparison to the potential of this class of molecule. The reason that there are so many there's so many potential avenues for proteins is fundamentally comes down to their sort of the structure or how they're made up. I'm sure that most of you will know that that proteins are made up of a string of amino acids, and each amino acid, there can be each position in the protein, there can be 20 different amino acids, and that creates this enormous combinatorial space, which as the protein gets longer and longer, the space starts to kind of reach ridiculously huge proportions. At the scale here, we're talking about 32 positions, which is a very small protein. You're already looking at 20 to the power of 32 different unique sequences, which is I think it's approaching the number of atoms that exist in the world. So it's an enormous space. And that gives you an enormous number of potential options when you're trying to discover a new protein. There are each time you add these, you change the different sequence, you change the length of the sequence, you change the composition, you get different shapes, and those different shapes lead to different functions. And so there are an enormous number of different potential applications for different proteins. And really, you could probably think of this for every single disease that exists, there's probably some kind of a corresponding protein therapeutic, whether that protein therapeutic is really simple, like blocking a specific biological interaction, or it could be a bit more complex, like engaging your immune system to combat a tumor. There are very, very many different potential applications for these kinds of proteins. However, that potential also makes the challenge of finding the right protein incredibly difficult. You have an almost infinite space that you have to search through in order to find the right protein for a given problem. And that problem is compounded because you're not looking for a protein that solves a specific problem. You're looking for a protein that solves many different problems. So you have to find, obviously, the first thing you have to do is to find a protein that actually does the thing you want it to do. But it isn't good enough to just have a protein that does the thing you want it to do. It also has to be safe. It can't be toxic. It can't be immunogenic. You need to be able to produce it at scale so that you can deliver it to all of the different patients that you want to treat. It needs to be stable so that it can survive when you're transporting it around the globe. There are loads and loads of different compounding variables that you have to factor in when you're discovering a new drug, and you have an infinite space to play with. So it's a really, really hard search problem. The way that conventionally this problem is tackled is really through that you end up searching for one or maybe two key properties of the protein, and that typically is the efficacy of the protein. So you search initially for a protein that does the job that you're interested in it doing. And then you filter, you have a bunch of different hits that come out of that process, and then you filter all of them down based on those other characteristics that you're interested in. So you check to see if they're manufacturable, you check to see if they're stable, you check to see if they're safe. And every single time you run those tests, you cut a bunch of proteins that fail. The problem with that is that you have it's an incredibly inefficient process. Often you might reach the point where you go through this process and you find that all of your proteins don't actually satisfy some of those requirements that you need for it to be a good drug. And then you have to go back to the start. You have to evolve a bunch of new proteins and see if any of those work. The approach that we're really trying to take with machine learning is to streamline that. So to find a way to simultaneously search for as many of these different properties as we can right from the start. And I'll explain how we want to do that using machine learning. Using machine learning really means that what we need to do this is to be able to produce a computational model for every single feature of interest. So for every single feature, we need some kind of a model which predicts the fitness of a protein based on its sequence. And in order to do that, we need to build those models from some kind of data. So I'm going to go into how we build those models now. We take this fundamentally an empirically driven approach. So the reason that we use machine learning as opposed to any other computational approach for building these models is that we tackle the problem of finding better drugs by basically testing them for the feature that we're interested in finding. And once we have that data, we then map that sequence back to the fitness of the molecules, and then we try and train a model based on that information. The way that we do that is through this process that I have on the screen here. So first of all, we produce a library of different sequences. So we have a starting point, we have a molecule which we think does something in the right scheme of what we're looking for the molecule to do in the end, and then we diversify it, and we create a bunch of different mutations in that gene. We synthesize those different mutations. We run a selection or some kind of screen of all of those different molecules to see how they perform. We sequence them. So then we take all of that data with, we take the assay data, we take and we sequence the underlying proteins, and use the scores that from those assays associated with those sequences to build machine learning models. once we have those machine learning models, we can then extrapolate to a much larger space. And basically, yeah, I can show you them on this slide here. So basically, the real benefit of taking this approach is that you can screen a relatively small area of sequence space, and those models then allow you to extrapolate to a much, much larger one. So in this example here, we screened around 5,000 different sequences for a specific for a specific feature. And then that allowed us to extrapolate to around 10 billion different sequences, and then extract, what were the very best sequences within that pool, with an accuracy of a Spearman R of about 0.8. So when we validated the sequences that were predicted to be good from this model, they turned out to be as we expected. Once you've done that, once you've built those individual models, you can then start to think about co-optimizing. So obviously I mentioned before that one of the key problems you face when you're developing new drugs is that you're not just looking to optimize a single feature, you're looking to optimize multiple. If you have a machine learning model which describes each of those individual features, you can then optimize across all of them to find the areas of sequence space which are shared in their particularly high fitness. So we use sort of a multi-objective optimization algorithm to basically fire a bunch of sequences at all of our machine learning models at once, and then optimize across all of them to find what is the sequence that is the best compromise across those different sets. And I'll give you some proper examples of how all of this works in a second. So that's the kind of the overarching concept of the platform we're trying to build. So now I'm going to kind of go into a bit more detail on a specific example of how we're doing this within a specific therapeutic area. So just to give you a bit of context on the therapeutic area, the problem we're trying to solve here is that in the space of inflammatory bowel disease, the clinically approved biologics, so protein drugs that are used, are administered via injection. And basically, those drugs are sort of anti-inflammatory biologics, and so when you inject them, they cause all sorts of systemic immunosuppression problems. So you end up with you're down-regulating the entire immune system, and because of that, you end up having all sorts of nasty side effects. And in addition to that, you also this is a chronic disease, and so people have to go into the clinic to have those injections every few weeks to keep this disease in check. What we are trying to do here is to design an orally available protein. So you can take this drug as a pill, and because you can take it as a pill, it solves two problems. The molecules are gut restricted, so they actually live in the site where the disease is. It's inflammatory bowel disease, so you want your anti-inflammatories to be in the gut. You don't want them to be everywhere else in the body. If you take them as a pill, then you can make sure that they only exist in that space, and then they're cleared from systemic circulation. So that has the potential to mean that these are safer, you have less side effects, and also more convenient because you don't have to, patients don't end up having to go into the hospital all the time to keep their injections going. The problem that you have to solve is that you're trying to develop a protein which survives in the gut, and the gut has evolved over many millions of years to destroy proteins. It's one of its purposes is to literally degrade them so that those proteins can be used, so that what you eat can be used, so the nutrients can be used by your body. And so there are all sorts of nasty enzymes, proteases, which degrade proteins in your gut. And what we're trying to evolve here is a molecule which is both potent, so it inhibits the immune response, but also it can survive in the context of those proteases. So how do we go about doing that? First of all, let's start with diversification. So initially we take a very systematic approach to this diversification. So this is just a very, very high level schematic. Obviously, this is not what our libraries look like, but we take an initial protein sequence, and we introduce mutations across that sequence. We we've tried a few different approaches to this initial phase of like evolving new proteins, and the one that we've ended up on is basically it's an entirely agnostic approach to the sequence itself. We don't care what the sequence is. We basically explore a local area around that sequence the same way every single time. And then based on the results of that small initial search, we'll build a much, much larger combinatorial library, which takes into account the findings of that initial screen. This is really, we found this to be an incredibly powerful technique. One of the problems that a lot of people face when they're doing directed evolution is that if you randomly mutate a gene, you'll find that the vast majority of sequences are worse than the starting point, and only a very, very small percentage are actually beneficial. And that represents a huge problem, because not only does it mean that you have a much, much smaller pool of sequences to work from, but also from a machine learning perspective, you just don't have very many data points to train, very many sort of positive data points to train your machine learning model on. Whereas the approach that we take is, as I mentioned, we have this systematic approach where we combine, we look at this initial search around the starting point, and then we throw in a bunch of the different interesting neutral beneficial mutations from that search, and we end up with this much, much higher performing library where the vast majority of sequences, over 80%, outperform the initial starting scaffold. Some of them massively outperform it, some of them outperform it a little bit, and some of them are worse. And again, that distribution is super important. You don't just want, you want to have as broad a distribution as you can, so that you can understand the underlying fitness landscape, the sort of the map of how sequences perform in this area of the space. So that's the first step in terms of diversification. So we screen, we build these libraries, and then I'm going to talk a little bit about how we actually generate the data. So once we've designed those libraries, we construct them, and this is obviously where a lot of the synthetic biology comes in. Traditional approaches might use random mutagenesis. They might use they might use a natural immune repertoire to search for improved sequences. We design all of our libraries in silico, and then we construct them synthetically. So we have our own internal library construction mechanisms for making these like high quality libraries, where we have like the vast majority of the sequences are high performing and high fidelity and that kind of thing. So that's kind of an important step there. We then screen them, and in this case, in this particular example, we're talking about potency. So we use a method called phage display, a pretty standard method in sort of drug discovery, protein drug discovery. You basically display each of the variants of the protein display on the outside of a phage capsid. The phage contains within it the DNA, and also obviously has the protein displayed on the outside. You can then basically select in a pool all of those different sequences for ones that bind to the target and those that don't. The difference comes in our process when we're thinking about how we actually run that phage display process. In a traditional directed evolution campaign, again, this is what people tend to do is they try to work through as much of the noise in that selection as they possibly can. So they'll run multiple panning rounds of these selections. So they'll continually clear out all of the sequences that don't work or what don't work as well, and they optimize for the very, very best performers. Then they'll take those best performers and they'll characterize them. They'll say, here are our hits. That's not how we do things, because ultimately what we want to do is to be able to build a model of this landscape. And building a model of the landscape means that you need to understand not just the very best performers, you need to also understand the space around them. And so we sort of adapt this phage display, the sort of standard phage display method to reduce the noise in that process as much as possible, but while also retaining the high and low performers. And we do that basically through next generation sequencing. So once we've run these selections, we sequence those pools of proteins, those pools of phages. We assign them scores based on how well they performed, based on how much they were enriched during the process. And then we do some validation. So we check at this point, does the stuff that we see in phage, those results we see in phage, do they correlate to some like meaningful cell-based potency assay? Just check there's a correlation there. So that's that first stage of actually producing this data. And then getting on to the building the models and refining them. We take the data from the next generation sequencing. Obviously, as I said, you have this very, very large pool of sequences that come from your phage selection. All of them have been sequenced. You've assigned some kind of a score to each of them based on how well they've performed. And then you have the data required to build a machine learning model. You have a sequence, you have a score, and then you throw that at a neural network and train it. And it will start to unpick what are the, where are the correlations in the sequence, and how does that relate to the function that you're seeing at the end. So once we build that machine learning model, we then do experimental validation. So we'll take the machine learning model, we'll say, here is what I think are the best sequences for this particular feature, be it in this case potency. We've never tested them; they're completely de novo. We synthesize those individually. We put them into our potency screen and see how they perform. And in this case, we get about 80% are about what we expected. So that's for one feature, and then we get into this question of multi-parameter optimization. Potency is not the only thing that we care about, right? What we care about is molecules that are both potent, but also can survive in the context of the gut. So we're not just running selections for potency or affinity, we're also running selections for stability. And we build models for both. And then we run a multi-objective optimization to find what are the best performing sequences that exist in this space. And we select some of those based on that. The best sequence, again, because this is multi-parameter optimization, there is no best sequence. It depends on what you're optimizing for. You might want to optimize more heavily for stability, or you might want to optimize more heavily for binding. That depends on the outcomes of your experiment. That depends on the kind of drug you're trying to develop. So we'll pick a range of those sequences that the model predicts are going to be great. We then put them into our assays and see how they do. And then this is ultimately the results of this project. Using this approach, we were able to improve the potency of these molecules by about 150-fold, while also massively improving their resistance against a whole different range of different proteases. So these are all proteases that exist within the gut that we determined would be important for a drug to be able to survive in the context of. We had, the starting molecule here is in purple, and this orange ring is what we were aiming for. And the green ring is the actual the characteristics of the proteins that we discovered using machine learning. And this molecule is obviously, it's the same molecule, so the potency data is in the same molecule as this green ring on the left here. So the molecules that we've developed in this project are now going into in vivo studies. So we're thinking about taking these into animal trials next and seeing how they perform in vivo. So that's kind of the case study on like how our platform works, and some sort of real data about showing how that it actually does what we say it does. I wanted to just talk a little bit about some of the core principles of building a machine learning platform. This is like something that I particularly find super interesting and is one of the things that I care a lot about. What we're trying to do is not just, produce a sort of a proof of concept piece of science which shows that we can apply machine learning to protein engineering. We're trying to build a platform which we can then exploit and use to generate loads and loads of different therapeutic molecules. And I guess I just wanted to go through some of the core principles that I think about a lot in my role about what we need to think about when we're trying to build a platform and not just build, a set of sort of bespoke scientific processes. Something that we can actually scale and produce many, many, many different therapeutics in the future. The first is something that we've kind of always really cared about at LabGenius, probably before it was even smart to do so, if I'm honest, which is putting standardization and automation at the heart of the science that we do. We hired our first automation engineer when we were probably about 8 people, when most of the science we were still figuring out. And that automation person kind of sat around like, why am I here? Like, I don't really understand, none of these experiments work yet. But on balance, I still think it was probably the right thing to do, because it's embedded automation as a key part of our culture as a company. And it's super important for us if we want to really build a platform for developing these kinds of molecules and using machine learning. Why? There are a few reasons. The first is reproducibility. A robot will always perform an experiment the same way every single time. It may have errors. It's not perfect. As much as I would love to say that like our robots never ever fail, they do sometimes fail. But at the very least, their failures are generally systematic, and if they do fail, they will tell us that they failed. That is like one of the problems that we see in science, and I'm sure everybody here will know, is that like reproducibility is a huge issue. The same person performing the same experiments in different labs can generate completely different data, and that data can it's incredibly difficult to unpick why that reproducibility is not there often. So you really want to fix as many of those variables as you possibly can, and automation and standardization. Standardization is like I guess step one, but automation is really nice in the sense that it enforces it in a way that's not really possible with standardization. When I talk about standardization, I mean like writing down exactly the protocol that the scientist uses when they run it in the lab, for example. The problem is that when a scientist has run that same protocol 100 times already, they might say, I know how to run this protocol now, or they might introduce little tweaks that they think might help in some way. That's obviously completely fine, and it's super useful to have that mindset of trying to improve protocols and change them. But when you're trying to really optimize for reproducibility, it can be incredibly damaging. So what we try and do is separate out work which we think is like core to the platform, things that we know work, and this is our versioned experiment. We run it the same way every time. It lives on a robot. You can't change that. You can't change that run without really getting a bit messy. From development work, where that kind of iterative improvement of protocols is much more like it's core to how we develop these things and how we make them work in the first place. The next thing that's super important about automation is that it enables a massive improvement in throughput. One of the experiments this is just a fun video where we were running the same experiment precisely in tandem with one another, not for any reason other than it looked cool, just to be clear, but it does look cool. And the benefits of throughput can be huge, because one of the things that we do after every single round of this process of machine learning driven, when we're sort of discovering these new molecules, is that we always want to validate them. And so that means, for any of you who've done protein expression and purification, it means basically producing a whole bunch of these proteins individually and then assaying them. And that process, doing that manually, can be incredibly time consuming. And through automation, we're now able to sort of produce hundreds of these proteins in parallel, which means that we can validate our models much more effectively, generate a lot more data, and then eventually, what we aim to do is to be able to produce different kinds of data, not just using, for example, phage display data, but also using free protein characteristics to be able to build machine learning models too. And you can only do that through massive scale, basically. The other thing, and this is, I guess, what most people think of when they think of automation, is it gives scientists back time. This is a little video that one of our automation scientists put together. We also have like some 3D printing stuff that goes on for like customizing individual bits of labware that we can't get our hands on or whatever. So we can just like print our own, just like do a bit of CAD and then like produce these little bits and pieces. And we now are in the position where like we can run these kinds of things, whether it's 3D printing or whether it's like actual experimentation. After everyone's gone home, they can just go press go, and the experiment will run overnight, and then you can come back in the morning and your results are there. So that's obviously a massive boon for scientists, because it means that you're not just spending your time in the lab using your hands all day. You can start to think about how making more use of the brains of the scientists. Ultimately, the reason that everybody gets into science is not to pipette colorless liquid from one well to another. It's because they want to discover new things, develop new processes, and that's the massive benefit of automation, is that it puts a lot of that time back into scientists' hands. The next thing is centralization of data. So one of the things that we have been doing a lot more of lately is thinking about how we can take all of the data that we ever produce in the lab and centralize it into one place. There are loads of benefits to doing this. I've put 3 down here. The first is that it really breaks down the barriers to collaboration and peer review. If traditionally when a lot of lab work is done, the results are sort of written down in a lab notebook with pen and paper, and the exact process that was used to generate that data lives in that lab notebook, and it's kind of inaccessible to anybody else who wants to have a look at it. If you basically democratize all of that data, put it all in one place where everybody can access it, it opens up this ability for everybody around your R&D team, everybody in your lab, to look at the data, understand it, review it, ask questions, and that is really where there's so much benefit in science. It's by getting the brains of different people on a particular problem. It also enables meta-analysis, which is really interesting. If you imagine that you're running these kinds of these cycles of directed evolution, and you're running them over months, over years, and the process is evolving over time. Things are changing. You're updating updating, improving processes, and you can start to look over time. If all of that data is stored in one place, you can see, okay, let's have a look at how this particular feature of an experiment has changed how it's influenced the data that we're producing over time. And understanding if something breaks, you can then go back and see, like, have a trace of like all of the things that have happened up until that point. And then finally, it also enables software automation. So one of the things that I've said, this diagram here is obviously super crude, but it's one of the things that we've been developing recently, which is now when anybody runs any experiment in the lab, all of the data is automatically pulled directly into a cloud, into somewhere in the cloud. And then once you have all of that data in one place, you can say to your software team, I need to run the same analysis on this kind of data every single time. And then they can say, okay, well, we'll write some automation so you don't have to do that anymore. So all of that data will go sucked into the cloud. You can come along and say, okay, cool, it's this kind of data. I need to analyze it in this way. It runs through an automated processing pipeline, and then your data comes out the other end, and it plonks it directly into your lab notebook where all of your experiment lives, and all of the data, all of the method lives. And that, again, it kind of has very much of the similar kinds of benefits of robotic automation. It gives scientists time back. It also means that the analysis of certain types of data is always done in exactly the same way, so you understand that, like, there isn't weird unexpected variation coming into your experimental processes. So yeah, these are the kinds of benefits that you can have from centralizing data. It's definitely not something that we have solved at LabGenius by any stretch of the imagination, but it's kind of one of the key principles that we take. And it's basically, we have a small internal software team, and it's basically their job at the moment to think about how we can just take all of the data that we generate and put it into one place so that it makes sense, so that we can automate a bunch of the work that happens on it and allow people to access it much more easily. And then finally, and this one cannot really be overstated, is to foster a culture of cross-disciplinary collaboration, openness, and learning. As I mentioned at the start, we have a team of very, very interdisciplinary, cross-disciplinary team. And the real kind of, I would say, the secret sauce of LabGenius is, like, obviously we have some really cool technology and some really interesting science that's going on, but all of that stems from the people. Ultimately, the people are what drive all of this innovation and drive the science. And the most exciting research and the most innovative research generally comes from getting people together from very different backgrounds and having them talk and trying to get them to talk in the same language. Because if you get too deep in your own area, you can often miss things, and it's at those interfaces of science where I think the real magic happens. So like combining data scientists with synthetic biologists, with software, with automation, there's so much exciting stuff that happens at that interface. And it's not easy. It doesn't happen just by putting everybody together in a room. You have to really like make this culture work, encourage it, foster it, and like put structures in place so that people talk in the right way, in the right language, and it's incredibly important to everything we do. It's also particularly within this field where we're actually thinking about how do we take these disparate fields of machine learning, synthetic biology. It's especially important here, but I think it applies to a lot of science where there's so much benefit to taking knowledge and expertise from different areas. So yeah, that's the end of my talk. Just to highlight that we're growing at the moment. We took on a bit more investment last year, and we're currently hiring. So if you are interested, check out our website. If there's nothing in particular that matches what you want to do at the moment, just like send an email over, and we can always be on the lookout for future roles and let you know when something else crops up. Particularly, yeah, I mean, all of the relevant fields of the people who are attending this talk, we're always kind of looking for people with the kinds of skill sets that you all have. So do check us out if you're interested. And yeah, that's it. So any questions? I guess I'm going to go to the chat. I don't know. I'm not exactly sure how this is going to work.
Yeah, no, that's great. Thank you very much, Harry. It's really interesting, the sort of technologies you guys are combining. So that was a really great talk. Thank you very much. Yeah, we do have a couple of questions. I'll go through the chat and read them out to you so everyone can hear it. If you do have any more questions, please do write them in the chat now, or just say you'd like to speak, and then you can unmute yourself and ask a question directly. But the first question is from Kelvin. He says, regarding the case study you gave, how different, homology-wise, is the final resulting target, i.e. the mutant, as compared to the starting point, e.g. the wild type?
So this is actually, it's a really interesting question. And the answer is, if I remember correctly, it's around, I think it's about 12 or 13 amino acids. One of the interesting things that we noted, I may even have a hidden slide on this, but I can't remember anyway, is that one of the really cool things about building the libraries and exploring the space in the way that we do is that it has actually enabled us to move a lot further from the wild type than if you were just exploring randomly, because you're already enriching for higher performing variants. That like drop off that you see when you get to a few positions away from the wild type it's longer. Like we've managed to sort of extend it outwards from using a sort of random mutagenesis approach, which is exciting. But I think, yeah, if my memory serves, it's about 12 amino acids. But obviously, yeah, something like that.
Very cool. Thank you very much. Raymond asks, you mentioned that centralizing data is an important part of your system. What data standards do you currently use?
Data standards, cool. So that's a kind of a complex question because it depends on the type of data that we're talking about. We have very, like, when it comes to NGS data, which is probably our most, so that's next generation sequencing. I'm not sure if everybody knows. We have pretty strict ways of storing and sort of structuring that data, because that's the data that feeds directly into machine learning. When it comes to centralizing data from sort of more conventional lab experiments, that is something that we're still working on. I wouldn't say that we have a strict data standard for like gel images, for example, but it's something that, again, it's really interesting how automation, both software and lab automation, can help enforce this kind of direction. If you have software automation which says the data needs to look this way in order for us to be able to process it correctly, it kind of like enforces this standardization of how your data should look when it comes out of an experiment, so that you can analyze it without having to without having to do it manually. So it very much depends on, as I said, very much depends on the data. For our NGS data, that's pretty much entirely standardized. It always looks the same way. For other data, it's still a work in progress, I would say.
All right, cool, thanks. Zoltan asks that there are two similar software packages, which I will not try and pronounce, that offer solutions for data centralization for SynBIC. Can you comment on why LabGenius decided to develop an in-house software versus buying it as software as a service?
Could you? I'm not sure exactly what you're referring to. Are you referring to a?
It's like TeslaGen and Riffin.
Okay, Riffin and TeslaGen. TeslaGen.
It's in the. I'm probably butchering that pronunciation.
No. That's I know both of those. So it's when you're looking at like taking software off the shelf, it's a constant battle of deciding this is going to save us a bunch of time, but it's always going to cost us in our ability to customize it to what the specific data that we're generating and how we want our specific data to look. And so it's something that we're constantly trading off of, like, how much of this should we just buy in versus how much of it should we develop ourselves. What we try to do is if there are things that are really standard, i.e. processes data that looks very much like any data that any other company would develop, we would buy some software to manage it. The example that I would probably give is, like, we use at least for, like, when we're doing cloning and things like that, we store all of our data in Benchling, because they have existing good tools where you can store sequences. It's easy to manipulate. All of that stuff is really good. But if it's other types of data, it's generally easier for us to just build a simple system using like Google Cloud Storage, where we can apply our own processing to it after we've generated it. So there's no one size fits all. It depends on the data. It depends on what it looks like, what we want to do to it afterwards, how we want to integrate it with other systems. It's always a question that we're constantly grappling with.
Okay, that's very interesting. Thank you for that. Dima asks, how do you approach privacy concerns? Since you centralize data, it seems to me that a large number of users have access to all sorts of data. Are there any restrictions of any sorts based on your role, etc.?
Yes, there are restrictions based on your role, but generally within the company, if you are a full-time employee, we don't restrict your access to data because the data we generate is not clinical data. It's not patient data. It's all in vitro experimental data. So there's no privacy concerns from that perspective. There are commercial confidence concerns, like, i.e. we don't want to just give access to somebody and then they take our data and like publish it on the internet. And for people who aren't full-time employees, or then we might restrict access. But we, as a general working principle, for data which is, as I say, all of the data we generate, it is not coming from people. It's just data that we ourselves have generated. As a general working principle, I don't like to restrict access to data based on who you are, because as I mentioned at the end there, one of the key principles of trying to build a company like this is to democratize data, is to allow people, regardless of who you are, what background you have, to be able to interrogate the work that we're doing and to be able to criticize it and think about how we can improve our process. If you start to say, well, this team gets access to this kind of data and that team gets access to that kind of data, you start to break down one of the key things that makes this kind of a company interesting to work for, and a lot of the benefits of having all of these kinds of different teams. So yeah, that's generally my thinking on
that. Okay, brilliant, thank you. I think Alyssa would like to ask a question. Are you able to unmute yourself?
Yeah, thanks. Can you hear me?
Yeah.
Yes.
Great. Well, yeah, first of all, thank you for a really excellent talk. So Yeah, this question, I understand if you can't comment on detail, but it'd be interesting to know. I was wondering, do you at LabGenius, do you limit your search to a particular type or fold or even size of proteins, or do you explore kind of a greater diversity?
Yeah, so we are, I'm trying to think what I can say. Basically, we start with a scaffold. So we start with a particular type of protein, and we work with a couple of different types of proteins, and then we evolve those for different mechanisms. So you could take, for example, an antibody. We would take an antibody and evolve that for a different purpose, whether it's improving its stability, improving its potency, changing its binding epitope, whatever that is. So generally, we start with like a functional protein and evolve it to do something different. We don't do any of that kind of de novo work where we're trying to like predict a particular fold or a particular structure. We try and abstract away as much of that kind of information to like the feature or the function that we're screening for. We'd like to incorporate more structural information though into our searches if we can, and that's something we're looking into. But right now it's very much like sequence to fitness is like, that's how we look at it. And then we restrict ourselves to particular types of proteins which do the kinds of things that we want them to do, and then sort of explore the local space around those.
Thanks.
Does that answer your question?
Helpful. Yeah, it does. Perfect. Thanks so much.
Cool. Cheers, Harry. Someone messaged me privately to say, will the slides be made available after this talk? I know the talk's being recorded, but are you able to provide the slides?
Yeah, it should be fine.
All right, sweet. Thank you very much. Next question. Okay, so this one's in two parts. I'll go for the first one first. For the synthesis part, instead of simply mutating the original protein, have you tried using ML techniques, e.g. k-nearest neighbors, to create protein sequences based on your current data set?
No, we have not tried that. So generally as I kind of mentioned during the talk, the machine learning doesn't feed into our library design so much as it feeds into just the predictions of high performing candidates. So the design we try and keep as broad as possible. Having said that, like, it's definitely something that's interesting. Like the more interesting ways that we can think about diversifying sequences, the better. So, but it's not something that we've looked into so far.
All right, sweet, thank you. And Lucas's second part was, it seems that protein sequences are like words in a text where little groups of letters are combined to make words and phrases. Have you tried natural language processing techniques in your project, e.g. recurrent neural networks, LSTM? I'm gonna add transformers on there as well.
Yeah, really, really great question. I am not the right person to answer that question, sadly. But I know that we have, it's the interesting thing about protein sequences and protein to fitness sequence of fitness landscapes is that often really simple, well, not really simple, but relatively simple functions actually describe those relationships pretty well. So in the case of if you're like looking to optimize the affinity or binding of a particular molecule, the complexity of that can be relatively simple. But then if you're looking to optimize the stability of the molecule, as I mentioned, like the complexity, you're talking about more of like some more complex structural changes to the protein, in which case the complexity of the function you use changes. So unfortunately, I can't answer your question specifically because I don't know the answer. To be completely honest with you, I'd need to get my my data scientist to have a chat. But yeah, I believe that we have tried it, but as I say, it very much depends on the feature that you're looking at. Sometimes really complex, interesting machine learning doesn't actually perform any better than a simple neural network. So. It very much depends.
Okay, I mean, I was going to ask actually then, does that mean you're, for most of your models, just using simple neural networks, or are you able to talk about that a bit more, just fully connected?
So as I say, I'm not the right person to ask. I know that we used feed-forward neural networks, convolutional neural networks to model some of these features, but in terms of like the detail, I'm afraid that I'm not the right person. I am but a humble wet lab biologist by training, and so I can't go into much depth.
It's part of being an interdisciplinary company.
Exactly. Yeah. It's like on my list is to like continue learning more about this along with everything else that we do. But unfortunately, I'm still pretty pretty ignorant when it comes to a lot of the data science that we do. To my shame.
Well, actually, on that topic, as someone who works at an AI/tech/biology company who comes from a biology background, a lot of the people in the audience here are, biology students or come from that sort of life sciences background and would really like to start getting into AI and stuff and machine learning. Is there any advice you could give to them, places to start?
That's a great question. There are some, it depends on what you want to get into. There are actually some really great textbooks around introductions to combining machine learning with biology and biotechnology, which I could send around afterwards. I can't remember the exact names of them off the top of my head. But if you want to get into it, I would recommend, okay, let me tell you what I know our lead data scientist would say, because which is, if you want to understand, if you want to really get into this stuff, it's important to understand the underlying statistics and maths behind all of it. A lot of people get into machine learning and they play around with training models based on some data that they can find, and that's fun for a while, and it can definitely get you into the area, just like playing around with some Python packages and things like that. But I know that like if you really want to get into it and you really want to get deep into it, getting into the statistics, getting into the maths is super important. That's not to say that like if that's really scary to you, which it is definitely for me, you should like stop yourself from doing some of those like those slightly lower hanging fruit just to get you into the area, like playing around with some data sets and building some models. But if you want to get deep into it, then I would recommend like boning up on your stats and really understanding what's going on behind the scenes.
Okay, great. I'm sure that's really encouraging to a lot of people here. So thank you very much. If there are any more questions, do make sure that you type them in the chat or you raise your hand. Raymond asks, hello, thank you for the talk. Your system seems really exciting. How similar/different is it to the Pac-Man prediction model? What type of reinforcement learning and ML techniques do underpin your framework? And a bit of a, obviously, you might not be able to answer the second part.
What kind I cannot answer the first part either, I'm afraid. One of our values is to be humble. I'm being humble today and telling you that I don't know the answer to your question. I'm sorry. I'm sorry about that. I don't know how it performs compared to that particular example.
So you've just talked to us about protein sequence today. Obviously, protein function to a large extent really is determined by its structure. And I also remember a long time ago, I looked on your careers page and you were looking for people with some structural bioinformatics skills in PyMol and whatnot. So I was wondering, does LabGenius work with any protein structural data at all, or are you just focused on trying to find the right sequence in that sequence space?
It's something that it's a great spot on our careers page. It's always a good way to find out about what companies are interested in doing. We are interested in incorporating more structural information into our models and into the way that we sort of generate our libraries and things like that. It's something that is something that we're interested in. It's not something that we do at the moment, and we have a relatively small team on our computational biology and bioinformatics side at the moment. But I know that it's something that we're interested in developing more on in the future. And so if that's something that you're like particularly is like particularly part of your skill set, then I would be happy to have a chat, because I know it's something that we want to build out capabilities in.
All right, sweet, thank you. On the topic of careers page, sorry if you already mentioned it, but a lot of the people in the audience will be undergraduates here looking for summer internships. Does LabGenius offer any of those?
Yes, absolutely. just like send an email to, Well, you can send an email to me. My email is harry@labgenie.us. Or I'm sure that there's a generic, I think there's a careers@labgenie.us account as well that you can send a message to. We're pretty much always open to people who are interested in running internships with us. So just let us know.
Okay, excellent. I think Raymond has a question to ask. Are you able to unmute yourself, Raymond?
Yes, hello. Thank you very much for the amazing talk. Very inspiring. I noticed that you presented the image of a very multidisciplinary team, and as a biomedical engineering, I really love that. What kind of systems do you have in place to foster collaborations between people from different disciplines?
Yeah, so there are a few things that we do. The first is that we have multidisciplinary teams. So when we run projects, we have functional groups. So people who, we have data scientist group, we have protein science, we have phage display groups. And obviously they all work together on the particular scientific area that they are specialists in. But then all of our projects are fundamentally multidisciplinary. And so we structure those around basically spanning all of those different teams and trying to focus everybody towards delivering on a specific objective for the company. And so everybody, should be as aligned as possible towards those objectives, as opposed to like the specific thing that's within their team. There are lots of other small things that we do. So that's kind of like the structural thing. But then every Friday we have different talks from anybody across the company, whether that's different areas of science, tech, business. People can give an hour's talk on whatever they want, and trying to obviously the emphasis being trying to make it as open to people from different disciplines as possible. We also have learning budgets, so like everybody gets I think £500 a year or something to spend on courses, to spend on books for any kind of learning that they want to do in different areas. We also support basically so that's basically trying to help people get into different areas that they want to get into, whether it's a biologist wanting to learn to code or a software engineer wanting to learn more about biology. So that is, I've lost my train of thought. And obviously we support basically people moving around within the company. Obviously you can't go directly from being a wet lab biologist to working in the data science team, but if that's something that you're interested in, we try and support that kind of growth as much as we possibly can. So again, giving you mentors within a different team, helping you to upskill in those different areas so that you can start to learn more about them. And I think generally, like the people who do the best at LabGenius, the people who kind of I guess like what am I trying to say here? I think that people who we really value, what we really value here is people who try to learn as much outside of their area as possible. And I think that's where you find the most value, not because necessarily you become a specialist in a different area that you started in, but it allows you to have conversations with people much more easily if you have at least the vocabulary of somebody working in a different field. And understanding a little bit of depth in that field allows you to have those conversations, which allows the kind of collaborations that we're talking about. So yeah.
That sounds wonderful. Thank you.
All right, brilliant. Thank you very much, Harry. It's really good to hear about all the support you give to your employees as well. So I think that's all the questions we're going to be getting this evening. Final thing to say before I hand off to Brandon is thank you very much, Harry, for your time. This evening, we really appreciate you coming, and best of luck to you and the rest of the guys at LabGenius.
Thanks so much.
To Brandon now.
Yeah, I guess that concludes our event. Thank you everyone for your attendance, and more importantly, for your incredible engagement. We hope to see you again soon in our future events. And a special thanks to our speaker, Harry, for the incredibly engaging and interesting talk today. We're excited to see where LabGenius takes this work next, and we hope to be able to hear from you again soon.
Thank you so much, everyone. It was really nice to chat, and thanks for all of the questions, including the ones that I couldn't answer.